Hi everyone, this is Rose again, and this is part 3 of the lecture on quantitative genetics. And the topic we'll discuss now is genetic variation. And I would like to start this part of the lecture with the key message from last lecture, which is that variation in quantitative traits is defined by many different loci, and each loci has its own small effect. Now, one way to analyze the differences between treatments or between groups would be by controlling the environment, like we do with laboratory mice. However, this kind of setup is very unpractical, and we have so much data from the real world. For example, like the Human Genome Project, there's so much information out there, but the environment is not controlled at all. So instead, what we do, we look at the variation of a trait. Now, how do we get to variation again? We need a couple of equations. This is the equation that we use to calculate the average. It's the sum of all the observations divided by the total number of observations. This is the equation for variance. So, for example, the phenotypic variance, which is the variation that you see in the phenotype. That is calculated by looking at xi, which is just one observation, minus the average, squared divided by the total number of observations minus 1. That is your variation. So basically what it is, it's kind of like a summary of the differences that you see in your population. And the standard deviation, that is simply the square root of the variation. So why is it that we are so interested by this variation and standard deviation and all this stuff that we usually find very boring because it has all these x's and y's and i's and Well, it's very useful because it brings us to the normal distribution. And the normal distribution is something that you will hear about a lot in science. And that is because many continuous traits are distributed like this. So what you have here on the x-axis, you will have your phenotype. So this can be height or milk production or whatever. And on the y-axis, it's the percentage. So what you see here is that the mean is called mu. If you do minus 1 standard deviation and plus 1 standard deviation, you'll see that 68% of your observations will fall within that range. And if you go the mean minus 2 times the standard deviation and the mean plus 2 times the standard deviation, you will see that 95% of your observations will fall within that range. Let's go to the example of the Western man. We knew that there was an average of 1.79 meters. And what I found was that the standard deviation is 8 centimeters. So now let's take a look at what is that area of 95%. How do we calculate this? We have a mean of 179. You simply do, when we go to the left, to 161 meter and 63 centimeters, we see that that is calculated by simply doing 179 minus 2 times 8. And then when we go to the right, 1 meter 95, same way but plus. So 179 plus 2 times 8. And this tells us that 95% of the observations falls, like of the observations, that being the height of man in the West, it will fall between 1.63 meters and 1.95 meters. And the other parts, those are the special men, the short guys and the tall guys. Now this bugger over here, this sigma, that is our phenotypic standard deviation. And let's go a little bit more into that. Our phenotypic standard deviation, if we square it, we get to the phenotypic variation. This phenotypic variance consists of the genetic variance and the environmental variance. And you find it simply by adding those two up together. Now let's take a look at the picture on the right. When we go to the top left, it shows the genotypic variation. Now what we see here is we have three genotypes. Small a, small a, big A, big A, big A. When we look within each genotype, so for example, we see here that within the small a genotype, the average is 3. But we see that the values vary from 1 to 5. When we go one graph down to the orange ones, we see that the average value is 5, but there's variation around that mean from 3 to 7. And when we go one graph down to the blue ones, we see that the average is 7, but that there's variation from 5 to 9. And what this so very greatly shows is that the small a genotype on average has smaller values than the big A genotype and the big A small a genotype. But within each genotype, there's also variation. So there's variation between genotypes and there's variation within genotypes. And that's when we blend those 4 graphs, the top ones, together into that final graph down there. And there you see that beautiful bell-shaped curve. When we look at variation in real life, in species, what we see is that when we look at differences between different lines of wheat, for example, the differences that we see are mainly genetic. They're due to genetics, because these are all these lines that have been selected for a specific something for a very long time. So they've been really genetically diverted. However, when we have a field with one kind of wheat, you will still see a lot of variation. You'll see short ones, you'll see big ones, you'll see average ones. And these differences are due to the environment. And for farmers, if somebody has a wheat farm or something, you know that it can be because of water, it can be because of light, it can be because of that in one part of your field there was a snail population that exploded and ate all your plants. So now let's take a look at this setup here. What we have is a small study to check how you can separate genetic and environmental variation. In this first generation, the parents, we have 2 purebred lines, and every single individual is homozygous. This means that there is 0 genetic variation in this population, in both of these populations. So that means that all the variation that we see within these lines are going to be due to the environment. When we cross those 2 purebred parents, the result will be the F1, and this F1 is going to be purely heterozygous. Again, they all have the exact same genotype, which means there is no genetic variation. There's only environmental variation. However, when we start to cross that F1, we will get the F2, and the F2 will actually have a whole bunch of different genotypes. And in this case, we have different genotypes in Hardy-Weinberg equilibrium, which means there is genetic variation. This means that our phenotypic variation is not just environmental variation, but also genetic variation. Now I would like to take you back again to this slide, where I say that the variation of quantitative traits is often determined by many different loci, each with a small tiny effect. Now let's take a closer look at this tiny effect. This is the tiny effect I'm talking about. It is the additive genetic variation explained by one small locus. And it's determined by 2 times the allele frequency for the one allele times the allele frequency of the other allele times the effect of one allele squared. Now let's take a look at how that variation is influenced by, for example, an allele frequency of 0. So imagine now that you have a p of 0. That would mean that all your alleles in the population would be the same. And that would mean that all the individuals have the same genotype, which means there is no genetic variation. And that means that for that specific locus, there is no genetic variation. And the same counts for when the effect size of that locus is of an allele is 0. If none of the alleles has an effect on the phenotype, then there's not going to be any genetic variation. Now, you don't have to know this equation for the exam, but it nicely illustrates how these P and Q and A influence the additive genetic variation. So now imagine we have these 6 loci here down here, and each of these locus has a small little additive genetic variation that they explain. Here's our total additive genetic variation. And altogether, if they're independent, they add up together to the total additive genetic variation. Then comes Jono with the question, but is all genetic variation additive? Nice one, Jono. No, it isn't. So here we have our equation. The phenotypic variation is explained by the genotypic variation and the environmental variation. Now let's take a closer look at the genetic variation. The genetic variation consists of additive genetic variation, also the dominance variation, and interaction variation. Additive genetic variation was this graph, where you see this nice and straight line. Dominance variation is when the heterozygotes acts a little bit differently than you would expect if it were additive. The interaction variation means, for example, that 2 genotypes interact, meaning that if you have 2 specific genotypes, like for example in the second graph, if you have B1 together with A2, you actually perform better than you would expect without interaction. Now let's go to the second part of this part of the lecture, which is about the interaction variation. So down here, we have the phenotypic variation. We say it consists of the genetic variation plus the environmental variation, and then there are 2 interaction terms. Right there. So let's start by looking at the genotype by environment interaction. And we'll use a little example with a potato plant. Imagine we have a potato plant and there's a locus, and the A1 genotype gives short roots, whereas the A2 genotype gives long roots. What we then see is when we look at 2 geographical locations, one in Ethiopia and one in Denmark. In Ethiopia, the A2 genotype, so that's the plants with the long roots, they outperform the plants with the short roots. And that could be explained because the ones with the long roots are better at reaching the water that's stored deep down in the ground. In Denmark, on the other hand, it's better to have short roots because there's enough water anyways. And this genotype by environment interaction is something that occurs in real life as well. Think about pig farming, freeland versus conventional. Same for poultry farming. And then there's the genotype by genotype interactions. Here on the left, you see this example where there is no interaction between the genotypes. Here you can see that if you have the B1 genotype together with the A2 genotype, you actually perform better than you would expect. In this situation, having both B1 and A2 actually makes you perform less.